Welcome to the Explainer. So, if you're looking to really understand the nuts and bolts of cellular biology, you already know that mapping protein DNA interactions is, well, it's absolutely fundamental. We're talking about unraveling those incredibly complex epigenetic frameworks that basically dictate how our cells operate. But historically, getting a clear, high-resolution map of these interactions has been a massive headache. It's tough. Today, though, we're focusing on a major methodological breakthrough that is literally changing the game for molecular biologists everywhere. It's called cut-and-tag sequencing.
Okay, let's dive right into our roadmap for today. We'll start with 1, the epigenetic research bottleneck. Then 2, enter cut-and-tag sequencing. Next up is 3, a streamlined experimental workflow, followed by 4, applications in basic epigenetics, and we will wrap things up with 5, CD genomics sequencing services. All right, starting with section 1, the epigenetic research bottleneck. If you've spent literally any time running traditional epigenetic assays at the bench, you know the daily grind all too well. We have a pretty stark contrast here between traditional ChIP-seq and what molecular biologists actually need to get their work done. Look, ChIP-seq has been the gold standard for a long time, but it comes with some seriously frustrating limitations. First off, it typically requires millions of cells. Millions. That makes studying rare cell populations practically impossible. It also demands a grueling 5 to 7 day experimental cycle. So yeah, you can go ahead and say goodbye to your weekends. It involves all this tedious formaldehyde cross-linking and ultrasonic fragmentation. And the kicker? Even after all that work, you're often battling this high background noise that completely obscures your data. So what's the ideal solution? Well, we need something that requires ultra-low cell input, has a drastically faster turnaround time, and actually delivers a pristine, high signal-to-noise ratio. Which brings us to section 2. Enter cut-and-tag sequencing. So what is this upgrade? Cut-and-tag stands for cleavage under targets and tagmentation. Basically, think of it as a highly precise molecular cut-and-paste machine. It's an antibody-guided technology that relies on an engineered TN5 transposase. Now, instead of just randomly fragmenting the entire genome with sonication and then crossing your fingers and hoping to fish out your target, cut-and-tag uses an antibody to zero right in on your specific protein of interest. Then, the TN5 transposase precisely cleaves the target DNA, and honestly, this is the genius part, it simultaneously adds sequencing adapters right at the binding site. Boom. You completely bypass the need for all that messy cross-linking and sonication. Now, what's really interesting about this slide is when we compare the numbers directly, the upgrade is just massive. For sample input, we drop from requiring millions of cells with ChIP-seq down to as few as 1,000 cells for a standard cut-and-tag assay. That's a huge difference. The procedure itself shifts from that harsh cross-linking over to direct adapter addition right in live cells or isolated nuclei. And the resolution? It tightens up dramatically. We move from these broad, clunky 1 kilobase regions down to a highly precise 200 to 500 base pair window. It's an absolute game changer in both efficiency and clarity. But honestly, perhaps the most shocking improvement is just this number right here, 1. By eliminating those cumbersome preparation steps we talked about, the entire experimental cycle drops from a highly variable 5 to 7 days down to just 1 single day. You go from a week-long marathon to a 1-day sprint, and you get cleaner data while doing it. No way around it. That's amazing. Moving on to section 3, a streamlined experimental workflow. So how exactly does this actually play out at the bench? Well, the mechanics function in 3 really elegant steps. First up is sample processing. This is where live cells or nuclei are gently permeabilized and a primary antibody is introduced to bind the target protein, which is then followed by a secondary antibody. Simple enough. Second is the transposition reaction. Here you add the protein A/G TN5 transposase complex, and it tethers right to the antibodies. Then, and this is super clever, you just add a buffer containing magnesium ions. That magnesium acts as a trigger. It activates the transposase to cut the DNA and insert the sequencing adapters locally. Finally, in step 3, library construction, you just amplify those purified fragments using standard PCR. That's it. There's no complex end repair or ligation required at all. Of course, once you have your sequencing reads, you pass them through a standard bioinformatics pipeline. You'll run quality control using tools like FastQC just to ensure your read quality is solid. Then you'll do some peak calling, often using Max2 or Seeker to separate your true specific binding signals from any lingering background noise, which generates what we call narrow peak files. Finally, you move right into functional annotation and visualization using software like ChIP-seq and IGV. That lets you associate those peaks with specific promoters or exons. Now, let's look at how this pipeline actually answers real biological questions. Which brings us to Section 4, Applications in Basic Epigenetics. One of the absolute most powerful applications for this is in histone modification analysis. For instance, researchers recently used CUT&Tag to map genome-wide histone modifications, specifically identifying marks like H3K27ME3 and H3K4ME3 across 7 distinct embryonic developmental stages in an aquatic model organism. And because CUT&Tag requires so little input material to work, they could finally analyze the true chromatin state dynamics of these early, super-fragile embryonic stages that were previously just completely off-limits to researchers. And the researchers noted something really remarkable from this. They stated, the dynamics of histone modifications were highly consistent with the timeline of transcriptome data, especially during ZGA, where chromatin opening and transcriptional activation were synchronized. This perfectly captures how CUT&Tag can reveal those synergistic underlying regulatory mechanisms that drive early cellular development and reprogramming. It connects the dots beautifully. Another core application here is identifying transcription factor binding sites. In another incredibly fascinating study, researchers used CUT&Tag to deeply analyze the chromatin binding characteristics of a specific factor known as LDB1. Because of that sky-high signal-to-noise ratio we mentioned earlier, they were able to precisely locate over 26,000 distinct binding peaks for the LDB1 factor across the genome in their specific cellular models. Over 26,000. And this brilliantly illustrates the power of that resolution. Thanks to the crisp data, they discovered that exactly 60% of these binding sites were located in enhancer-related regions, like intergenic or intronic spaces, while the remaining 40% were concentrated directly in promoter regions. Insights like this allow researchers to accurately map out exactly where and how core transcriptional regulatory circuits are forming on the genome. So, seeing these really clear, high-resolution results naturally brings up an important question for any focused researcher watching. Are you ready to upgrade your epigenetic research? And honestly, more importantly, why on earth would you struggle to optimize this complex bioinformatics and wet lab workflow internally when you really don't have to build the whole pipeline from scratch? Which leads us perfectly into Section 5, CD Genomics Sequencing Services. Look, if you want the pristine results of CUT&Tag without the massive headache of initial optimization, you need an expert partner. That's just a fact. CD Genomics offers a high-quality CUT&Tag sequencing service that is totally dedicated to precise protein DNA interaction mapping. They are positioned as the ultimate provider of state-of-the-art platforms for both academia and industry. By leveraging their specialized service, you essentially get to bypass all the technical limitations and those steep learning curves that always come with adopting a brand new molecular protocol in-house. And remember earlier when we said CUT&Tag standardizes at around 1,000 cells? Well, get this. CD Genomics has optimized their proprietary workflow even further. They offer an absolutely incredible ultra-low cell input capability. For certain sample types, they can drop the requirement down to as few as 60 cells, just 60, and they still deliver highly accurate, reproducible, and low background results. That is totally game-changing for anyone working with incredibly rare or really difficult to isolate cell types. Plus, they don't just, hand you a hard drive full of raw sequencing reads and say, good luck. CD Genomics provides really robust end-to-end bioinformatics support. Their pipeline covers everything from the basic genome alignment and peak calling to advanced motif analysis for transcription factors, gene ontology analysis, pathway analysis, and even differential peak analysis. They basically translate all that raw biochemical data into highly actionable visual insights for your specific gene regulatory networks. So the crucial point is knowing exactly what you need to do to get started. If you look at their practical guidelines for sample submission, it's actually very straightforward. If you're submitting cells, they generally recommend aiming for greater than 85% viability post-thaw, with cell counts tailored to whether you are targeting general proteins or specific transcription factors. If you are submitting tissue instead, the guidelines are just based on sample weight. It is a highly structured, super researcher-friendly onboarding process. So, to sum up our explainer today, the shift from traditional ChIP-seq to CUT&Tag is not just some small incremental improvement. It is a fundamental upgrade in how we map the epigenome. And by partnering with experts like CD Genomics, you can instantly master data analysis and result interpretation, walking away with a proven high-resolution chromatin interaction map that will absolutely drive your research forward faster. I'm going to leave you with this final thought to ponder today. As you look at the fundamental cellular processes you study in the lab every day, what hidden epigenetic mechanisms will you uncover when you finally eliminate that background noise and those frustrating sample limitations? The tools are ready and waiting for you. Thank you so much for joining me on this deep dive into cut-and-tag sequencing. Keep questioning, keep learning, and I will definitely see you in the next explainer.